Welcome to the podcast series The New Student Pharmacist, where we discuss chemistry and pharmacy, as well as leaders in pharmacy careers, community, and chemistry and pharmacy research. We encourage you to support the work we are doing and follow us on Spotify and Apple Podcasts by subscribing for free. Note, the views on the podcast represent those of my guest and I.
Purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I.
Welcome to The New Chemist. We're glad you're listening. Feel free to download this podcast on Apple Podcasts, Google Podcasts, and Spotify. Here on The New Chemist, we discuss chemistry, which simply put is the science of change, as well as careers, community, research, and COVID-19. We're happy you're tuning in. My guest today is Dr. Robert Langer. Thanks for joining me today. It is good to hear from you. Just briefly, I'll inform my audience about you. Dr. Robert Samuel Langer Jr. is an American chemical engineer, scientist, entrepreneur, inventor, and one of the 12 Institute professors at the Massachusetts Institute of Technology. He was formerly the Germershausen Professor of Chemical and Biomedical Engineering and maintains activity in the Department of Chemical Engineering and the Department of Biological Engineering at MIT. He is also a faculty member of the Harvard-MIT Program in Health Sciences and Technology and the Koch Institute for Integrative Cancer Research. In 2015, Dr. Robert Langer was awarded the Queen Elizabeth Prize for Engineering. He has numerous accolades and achievements. Please welcome Dr. Robert Langer.
Dr. Langer's influential work has garnered him over 220 major awards. Among these are both the United States National Medal of Science and the United States National Medal of Technology and Innovation. His contributions to the field have been acknowledged through a vast array of other awards, including the Gardner Foundational Award, the Charles Stark Draper Prize, often likened to the Nobel Prize for Engineers, the Millennium Prize, the Priestley Medal, the Wolf Prize in Chemistry, and the Kyoto Prize. In 2015, Dr. Langer was the recipient of the prestigious Queen Elizabeth Prize for Engineering. Dr. Langer's research is at the cutting edge of biotechnology and materials science, focusing on the development of polymers for the controlled delivery of drugs, as well as the engineering of new tissues. His academic achievements are matched by his role as a member of multiple national science academies and his receipt of over 30 honorary doctorates from top-tier institutions worldwide. Very accomplished engineer with over 400,000 citations. His influence extends beyond academia into the realms of biotechnology and global health. Forbes and BioWorld have highlighted him as one of the most pivotal figures in biotechnology, and Time Magazine and CNN have recognized him as one of the most significant people in American science and medicine. It is definitely a treat to have Bob with us today, and it's good to have others joining in this informal mentorship meeting. Ugo, it's good to see you. So, drug delivery. We're just going to go through a few basic ideas in this presentation, and we will proceed to discuss some as associated with some of the work that Dr. Langer did. The drug delivery systems, according to the National Institute of Biomedical Engineering and Bioengineering, describe technologies that carry drugs into or throughout the body. So if we do like a quick overview, we've gone from ancient remedies and herbal medicines through the pharmaceutical revolution to oral dosage forms, to transdermal patches and biotechnology and gene therapy, to targeted drug delivery and smart drug delivery systems. So you have targeted drug delivery, biocompatibility and safety, controlled release systems, nanotechnology and nanomedicine, and personalized medicine, which is a really hot topic now. So some of Dr. Langer's achievements: Bob has achieved a number of things. Some of his work has been centered around investigating the mechanisms of release from polymeric delivery systems with concomitant microstructural analysis and mathematical modeling, studying and applications of these systems, including the development of effective long-term delivery systems for insulin, anti-cancer drugs, growth factors, gene therapy agents, and vaccines, developing controlled release systems that can be magnetically, ultrasonically, or enzymatically triggered to increase rates, and angiogenesis inhibition. Done a lot of work. So some of the big themes that kind of encapsulate the work that he's done: polymeric delivery systems, controlled release systems, angiogenesis inhibition, biodegradable polymeric delivery systems, tissue engineering and biomaterials, and some key questions. So this is going to be a dialogue. So some key questions that we have for you, Bob, are: how did the work that you did help with the research you're doing now in drug delivery? Once again, it's good to have everyone on today.
Yeah. Well, so really, the way I got started in drug delivery was by doing cancer research, trying to have a delivery system that I could use to deliver large molecules, which hadn't been done before, from tiny particles. So that, and of course, at the time I did this was first published in Nature in 1976. I don't think there were very many large molecules that were of interest. Today, of course, there are many, and drug delivery has been key to that. I mean, not only for delivering, proteins, but, the recent work that, again, one of the companies, as some of you know, that I started was Moderna, but the idea that you could deliver messenger RNA if you didn't have the mRNA, which is a large molecule, and nanoparticles, we wouldn't have the COVID vaccine, and I believe many other treatments that will come in the future. And of course, that is some of the things that we're doing today in drug delivery. We're working on delivering complex molecules by many routes: oral, through swallowable, through nanoparticles, through other routes as well.
Yeah, so we see that it advanced the field in certain areas. However, what were the lasting impacts made during the 70s, 80s, and 90s in your experience and opinion? During your early years of this work, what would you say were the lasting impacts that you saw then and that you are seeing now?
Well, I think the first impact was that you could do it at all. A lot of people didn't think you could deliver large molecules. In fact, the work we did when we wrote grants was rejected many times, and a lot of people who were experts just said that this was impossible, like trying to get a large molecule out of a tiny particle. They said either it would come out instantaneously or it wouldn't do it at all. And so, I think that was really the first thing. But then work by us and also by others, would lead to many things that I do think have had an impact. First, it's basic science, trying to understand the mechanisms by which you can get delivery, trying to create new materials, trying to get the right kind of kinetics, different types of design. So many different things were done in the 70s, 80s, and 90s. And of course, I was fortunate. I had a lot of very great students and postdoctoral fellows who are now like professors or involved in companies all over the world. And, they did a lot of that work. And then there were lots of other people that I knew that, got involved in this as well.
Okay, sounds good. So just before you proceed, I'll open the floor for any questions. Any questions that people have, I'll open the floor for questions, and then we'll proceed with the presentation.
Dr. Langer, I do have a question.
And you should just call me Bob. All of you should call me Bob.
All right. Wait, no, you know that I'm from the old school.
Well, you're an important person. You sir, all of you are.
Well, listen. What are your thoughts on now? You talked about nanoparticles, and typically the nanoparticles in the past were created using lysosomes, right?
Liposomes, yes.
Liposomes. What are your thoughts on niosomes now?
Well, I think it's a good question. I mean, so people are making nanoparticles out of all kinds of things over the years. Whether it's lipids or polymers or really anything. I think all of them are interesting scientifically. I think the issues that I would think about would be, what I always call do they have some unfair advantage for a particular application compared to what already exists? And, it might depend on the particular liposome or niosome or polymerisome or whatever it is that you have to do. So I don't know that there's a simple answer. And people may come up with ways of making them better and better. Interestingly, when I started doing this, a lot of people also felt that lipid nanoparticles or liposomes were a nice academic thing, but they would never really be useful clinically. And clearly that's changed because people figured out better and better ways to make them and better components. So, and I think that's one of the great things about science. You can keep evolving it and making it better and better. So I always hesitate to say what's better. I think all these things are good, and can have potential advantages.
Okay, so go ahead, Dr. Tin. Do you have anything else you want to ask?
No, we'll let that go.
Mm-hmm. Okay, yeah, that's good. Okay, so moving right along. The presentation was composed based off of the extended list of articles that I found for that Dr. Langer played a part in writing or was an author in. So they were organized by years and segmented by each 100 in terms of how it was arranged in the list, and the themes highlighted that covered the ideas. So if you look at the early portion of Dr. Langer's of Bob's work, we see that many principles, technologies, and concepts he developed during the 1970s and 1980s continue to be relevant and are actively applied in the design of modern drug delivery systems with a focus on improving therapeutic efficacy and patient outcomes. So some themes, so these are from some of his Bob's early work. So you had enzymatic regeneration of ATP in 1974 and 1976. Then you went on to the pH dependence of assays, mast cells and tumor angiogenesis, polymers for sustained release in 1976, the isolation of a cartilage factor in 1976, and continuous long-term intraarterial infusion in 1978. Moving right along, his work then proceeded into the controlled release of macromolecules for polymers in 1980, zero water release kinetics in 1980, implantable drug delivery systems in 1981, microspares for controlled drug delivery in 1984, and local controlled release of diphosphonate in 1985. So, the question posed to Bob, you is, would you say that your work with Judah Folkman really laid the foundation for you to do this work that you did in the early portion of your career? Or would you say it was your graduate work and your postdoctoral work with Judah Folkman that complemented to the progress, advancement, and drug delivery research milestones during this time?
Yeah, I would say that it was definitely the work with Judah Folkman. I mean, he, partly because of trying to solve the angiogenesis problem, and partly, because, and then the work with him, and then the later subsequent work at MIT that I did with different graduate students. But that was really the key things.
Okay, sounds good. Okay, moving right along. So the questions that we have, what were the foundational principles that guided your research inquiry during the 1970s and 1980s? What were the overarching questions that you were trying to answer in the 1970s and 80s? And are they still influencing your current practice in the field?
Yeah, so I would say that, part of it, it was trying to understand, since people told us we couldn't deliver large molecules, and I felt we were doing it, there were a couple things. One was doing it and doing it very reproducibly. A second was truly trying to understand the mechanisms. A third were developing newer and better materials. And I'd say some of those I feel we've solved, but they still influence what I do today. I mean, we continue to try to figure out, how to make better systems and solve new generations of problems. I mean, now that we have the foundation to make nanoparticles or microparticles or just deliver large molecules in general, you always want to do better, right? Can you target them to specific cells? Can you cross barriers in the body that are tough to cross, like the intestine or the brain? But I think the foundational principles were.
But any thoughts on that, Aaron?
Yeah, absolutely. Well, one of the ones. I mean, there are other drugs like Alkermes, which was based on some of our work. They've been using, I think it's now naloxone or naltrexone. But you're right about buprenorphine. I mean that right now is like in a sublingual system. And I believe some people are developing on, injectable ones, and maybe that's what you meant, where they're sub-Q or intramuscular. I mean, I'm not an expert. I don't know what all the issues are. I mean, I think some of the things that I know people have talked about are, could you make them last longer? I think that's certainly possible. I know I talked once with Richard Pops, who was the CEO of Alkermes, which is one of the producers of the naltrexone one, and he said, that's a 1 month system. They'd like to make maybe someday a 3 month system or a 6 month system. So I think developing them longer probably would be, one of the things that would be useful. That particular system is called Vivitrol. But, and I would think that those kinds of principles could be extended to buprenorphine, but I'm not expert enough to know all the issues with buprenorphine.
All right. Vivitrol has worked well. I still think that there are some challenges with therapeutic concentration and maintaining that therapeutic concentration.
Making it flatter, you mean?
Yeah.
Yeah. Well, that's a good question, and that I think is something that could be done, by, I actually mentioned some ways long ago. I was actually on their board, maybe 20 years ago, and I agree with, I think the ways to do it. You could, if you make sort of the effective particle size very, very tiny, I think you could probably help on that. And the other thing, though, maybe it's not a good idea from a manufacturing standpoint.
I am very grateful for the excellent role model, leader and trailblazer I have had the opportunity to interact with, receive advice from and work with at points. Dr. Robert Langer, MIT Institute professor and the co-founder of Moderna. in a celebration of the work he has done and the accomplishments he has made in drug delivery. I was able to find a list of some of his citations, namely 1,561 of them. So if you want to find the citations which we will discuss, you can use Google Scholar. Compose a list of his citations where he is listed as an author, arrange them by years, and then using the theme you can find the papers relevant to the themes which will be discussed in this episode. So here is the beginning of the episode. This episode will discuss his citations based off of ranges by the hundred and years in ascending order. With the use of software tools, Google Scholar and other resources, this audio review was composed. Episode title: Ode to 1,561 Citations. References 1 to 100. The references provided cover a wide range of research conducted by Robert Langer in the field of drug delivery and related areas during the 1970s and 1980s. While these studies are several decades old, they have had a profound impact on the field of drug delivery, and many of the principles and technologies developed during that time continue to be relevant today. Here are some implications and contributions of these references to drug delivery today. 1. Enzymatic regeneration of ATP, 1974, 1976. The study of enzymatic processes for regenerating ATP has implications for the development of drug delivery systems that rely on energy sources, such as implantable drug pumps or controlled release systems. Understanding the kinetics and thermodynamics of these processes can inform the design of more efficient and long-lasting drug delivery devices. 2. pH dependence of assays, 1976. Knowledge of pH-dependent assays is important for drug formulation and development, as it helps in optimizing drug stability and release under various physiological conditions. This is particularly relevant for oral drug delivery systems, where drugs may encounter different pH levels in the gastrointestinal tract. 3. Mast cells and tumor angiogenesis, 1976. Understanding the role of mast cells in angiogenesis can have implications for the development of targeted therapies to inhibit tumor growth by controlling angiogenesis. Drug delivery systems can be designed to deliver anti-angiogenic agents directly to tumor sites. 4. Polymers for sustained release, 1976. Langer's work on polymers for sustained release of proteins and macromolecules laid the foundation for modern drug delivery systems. Today, biodegradable and non-biodegradable polymer-based drug delivery systems are widely used for controlled release of drugs and biologics. 5. Isolation of a cartilage factor, 1976. This research has implications for the development of drugs or factors that can inhibit tumor neovascularization. Drug delivery systems can be employed to deliver these factors directly to the target tissue or tumor site. 6. Continuous long-term intra-arterial infusion, 1978. This work is relevant to the development of continuous drug infusion systems used in various medical applications, including cancer therapy and pain management. 7. Controlled release of macromolecules from polymers, 1980. Langer's review on controlled release from polymers is a seminal work in the field of drug delivery. It highlights the importance of sustained drug release for improving therapeutic outcomes and reducing side effects. 8. Zero-order release kinetics, 1980. Achieving zero-order release kinetics from drug delivery systems is a critical concept that continues to be explored and applied in the design of controlled release systems for various drugs and biologics. 9. Implantable drug delivery systems, 1981. The development of implantable drug delivery systems, discussed in several references, has paved the way for long-term drug delivery solutions, including subcutaneous implants and drug-eluting devices used today. 10. Microspheres for controlled drug delivery, 1984. The use of polymeric microspheres for controlled drug delivery has become a well-established approach in the field of drug delivery. These microspheres can be tailored to release drugs at a controlled rate, offering prolonged therapeutic effects. 11. Local controlled release of diphosphonate. 1985. The application of controlled release systems for preventing calcification of bioprosthetic heart valves is an example of how drug delivery can be used to enhance the performance and longevity of medical implants. In summary, Robert Langer's pioneering work in drug delivery has had a lasting impact on the field. Many of the principles, technologies, and concepts he developed during the 1970s and 1980s continue to be relevant and are actively applied in the design of modern drug delivery systems, with a focus on improving therapeutic efficacy and patient outcomes. References 101200. The references provided cover a wide range of topics related to drug delivery and biomaterials. Here are some implications for drug delivery today based on the information from these references. 1. Magnetically controlled release systems. Reference 101. The use of magnetically controlled drug delivery systems could have applications in targeted drug delivery. This technology could allow for precise control over the release of drugs at specific sites within the body, potentially reducing side effects and improving therapeutic outcomes. 2. Bioerodible polyanhydrides. Reference 102. Bioerodible polymers continue to be a significant area of research for drug delivery. These polymers can be used as drug carriers, and their degradation characteristics can be tailored to control drug release over time. Today, researchers are exploring advanced formulations and applications of bioerodible polymers for various drugs. 3. Enzymatic control of anticoagulation. Reference 104. Enzymatic control of anticoagulation remains a relevant topic in medical device development, especially for devices like artificial hearts and extracorporeal circulation systems. Techniques for controlling anticoagulation enzymatically can improve the safety and performance of these devices. 4. Heparin research. References 105, 130. Heparin and related compounds continue to be essential in anticoagulation and other medical applications. Research on heparin structure and activity is crucial for developing safer and more effective anticoagulation therapies. 5. Local controlled release for heart valve calcification. Reference 106. Localized drug delivery to prevent calcification in bioprosthetic heart valves remains an important area of study. The development of controlled release systems for this purpose could improve the longevity and performance of heart valve implants. 6. Enzymatic treatment for neonatal jaundice. References 107, 124. Enzymatic treatment for neonatal jaundice is an example of how immobilized enzyme reactors can be used therapeutically. Such systems may continue to find applications in neonatal care and other medical fields. 7. Controlled release of macromolecules. Reference 108. Controlled release of macromolecules is still a vital area of research, especially in the context of biopharmaceuticals and gene therapies. Developing systems that can provide sustained and controlled release of these large molecules is critical for their clinical success. 8. Biodegradable polyanhydrides. References 111, 167. Biodegradable polymers, including polyanhydrides, are widely used in drug delivery systems. Their biocompatibility and controlled degradation properties make them valuable for delivering a variety of drugs, including peptides and proteins. 9. Polymeric controlled drug delivery. Reference 183. Polymeric controlled drug delivery systems continue to evolve with advancements in polymer science and nanotechnology. These systems offer precise control over drug release kinetics and can be tailored to various therapeutic applications. 10 Ultrasound Enhanced Polymer Degradation. Reference 197. The use of ultrasound to enhance polymer degradation and drug release is an area of ongoing research. This technology could lead to innovative approaches for on-demand drug delivery. 11 Immobilized enzyme reactors. References 107, 124, 155. Immobilized enzyme reactors have a wide range of applications, including detoxification and drug synthesis. These systems are valuable in biotechnology and bioprocessing. 12 Biocompatible materials. References 175, 200. Biocompatible materials, such as bioabsorbable polymers, are essential for tissue engineering and regenerative medicine. They enable the development of implants and drug delivery systems that interact safely with biological systems. In summary, the research and insights presented in these references from the 1980s have paved the way for numerous advancements in drug delivery and biomaterials. Today, researchers continue to build on this foundation, developing innovative drug delivery technologies with the potential to improve patient outcomes and revolutionize the field of medicine. References 201 to 300. The references provided cover various aspects of drug delivery and related research from the late 1980s to the early 1990s. Here are some implications and key findings from these references. 1. Bilirubin detoxification. Reference 201 discusses the simulation of bilirubin detoxification in newborns using an extracorporeal bilirubin oxidase reactor. This research likely contributes to the development of medical devices or systems for treating newborns with jaundice, improving their health outcomes. 2. Heparinase immobilization. Reference 202 discusses heparinase immobilization, which could have implications in the development of improved heparinase-based therapies for patients with blood clotting disorders. 3. Transport rates of proteins. Reference 203 explores the transport rates of proteins in porous materials with known microgeometry. This research may be relevant for designing drug delivery systems with controlled release rates and improved drug distribution within tissues. 4. Controlled release of dopamine. References 204 and 205 describe the controlled release of dopamine from polymeric brain implants. This research could have implications for the treatment of neurological disorders such as Parkinson's disease through sustained drug delivery. 5. Poly-anhydride microsphere morphology. Reference 206 discusses the morphology of poly-anhydride microsphere delivery systems. This research may contribute to the design of drug delivery systems with specific release properties. 6. Hydrolytically degradable polymers. Reference 207 discusses hydrolytically degradable amino acid containing polymers. Such polymers may find applications in drug delivery systems designed to degrade over time, releasing drugs gradually. 7. Microencapsulated liposomes. Reference 208 explores microencapsulated liposomes for enzymatically controlled pulsatile release of biologically active substances. This technology could have applications in drug delivery systems requiring precise control over drug release. 8. Inhibitor of neovascularization. Reference 209 identifies an inhibitor of neovascularization from cartilage. This discovery may have implications in anticancer and anti-angiogenesis therapies. 9. Medical reactors. Reference 210 discusses medical reactors, which might be relevant for the development of medical devices and systems for drug delivery and other applications. 10. Biocompatibility of polymeric materials. references 214 and 215 delve into toxicology and biocompatibility considerations in the evaluation of polymeric materials for biomedical applications. This research is crucial for ensuring the safety of medical devices and drug delivery systems. 11 Polyanhydrides as drug delivery systems. Reference 215 explores polyanhydrides as drug delivery systems. Polyanhydrides have been studied extensively for their potential in controlled drug release. 12 Tissue engineering. References 216, 240, and 241 discuss tissue engineering using polymeric substrates and constructs. These findings may have applications in regenerative medicine and tissue replacement therapies. 13 Ultrasound-enhanced drug delivery. References 227 and 288 mention ultrasound-enhanced drug delivery, which could lead to non-invasive methods for improving drug penetration and release. 14 Protein formulations. Reference 270 focuses on protein formulations for animal health applications, which may have implications for veterinary medicine and pharmaceuticals. 15 Insulin aggregation. Reference 284 investigates insulin aggregation and stabilization, potentially contributing to the development of more stable insulin formulations for diabetes treatment. 16 Responsive polymer systems. References 231 and 275 discuss responsive polymer systems for controlled drug delivery. Such systems can provide on-demand drug release, enhancing treatment efficacy. These references collectively demonstrate the diverse research efforts in drug delivery, biomaterials, and medical device development during the late 1980s and early 1990s, with potential applications spanning a wide range of medical fields. Researchers during this period were exploring innovative approaches to drug delivery and biocompatible materials, laying the foundation for advancements in healthcare. References 301 to 400. The references provided cover a wide range of research related to drug delivery and biomaterials from the early 1990s. Here are some implications for drug delivery today based on the information from these references. 1. Polymers for controlled drug delivery. The references discuss the use of polymers as controlled drug delivery devices. Today, this concept has evolved, and researchers continue to explore new polymer-based drug delivery systems. These systems offer the advantage of precise control over drug release, reducing the frequency of administration and improving patient compliance. 2. Tissue engineering. Several references touch upon tissue engineering using biodegradable polymers. This field has seen significant advancements in recent years, with researchers working on creating functional tissues and organs. The implications for drug delivery include the potential for targeted drug delivery within engineered tissues or organs. 3. Enzymatic modification. Reference 305 mentions enzymatic modification of plasma low-density lipoproteins as a potential treatment for hypercholesterolemia. Enzyme-based therapies have continued to develop, including enzyme replacement therapies and targeted enzyme delivery systems for various diseases. 4. Microspheres for drug delivery. References 318 and 395 discuss the use of microspheres for controlled drug delivery. Today, microsphere-based drug delivery systems are extensively researched, and they offer various advantages, such as prolonged drug release and localized drug delivery. 5. Polyanhydrides. References 306, 343, and 394 mention the use of polyanhydrides for drug delivery and tissue engineering. Polyanhydrides have remained an area of interest, and they have been explored for applications like vaccine delivery and sustained drug release. 6. Electroporation for drug delivery. References 313, 330, and 331 discuss electroporation as a method to enhance drug delivery. Electroporation continues to be explored as a non-invasive technique for transdermal and intracellular drug delivery. 7. Biodegradable scaffolds. References 333, 377, 380, and 381 mention the use of biodegradable scaffolds for various applications, including liver tissue engineering and cartilage regeneration. Today, biodegradable scaffolds are used in tissue engineering and regenerative medicine to provide structural support and controlled drug delivery to aid in tissue repair. 8. Nanoparticles. Although not explicitly mentioned in your references, nanoparticles have become a significant focus in drug delivery research. Nanoparticles offer a versatile platform for drug delivery due to their small size, which allows for targeted delivery and controlled release. 9. Personalized medicine. Advances in drug delivery have paved the way for personalized medicine. Researchers are exploring the customization of drug delivery systems to match individual patient profiles, which can improve treatment outcomes and reduce side effects. 10. Clinical translation. The research mentioned in these references laid the foundation for many drug delivery technologies that are now being clinically tested and translated into practical medical applications. The continued development of these technologies holds great promise for improving patient care. In summary, the research presented in these references has had a lasting impact on the field of drug delivery. Today, drug delivery systems are becoming increasingly sophisticated, enabling more precise and effective treatment strategies across a wide range of diseases and conditions. References 401 to 500. The references provided are from 1995 and 1996 and cover a wide range of topics related to drug delivery, biomaterials, and tissue engineering. Here are some of the implications and highlights from these references for drug delivery today. 1. Electroporation for drug delivery. Reference 402. The use of electroporation for transdermal transport of DNA antisense oligonucleotides was explored. Electroporation is still a relevant technique for enhancing drug delivery across the skin, and it has applications in gene therapy and vaccine delivery. 2. Polymeric drug delivery systems. References 463 and 490. These references discuss the development and characterization of polymeric drug delivery systems. Today, polymer-based drug delivery systems continue to be a significant area of research and development, with applications ranging from sustained drug release to targeted drug delivery. 3. Tissue engineering. References 407, 408, 422, 441, 452, 481. Several references touch on tissue engineering, including the fabrication of tissue-engineered organs and the use of biodegradable polymers. Tissue engineering has evolved significantly since then and holds promise for regenerative medicine, including drug delivery within engineered tissues. 4. Ultrasound-mediated drug delivery. Reference 404. Ultrasound-mediated transdermal protein delivery was explored. Today, ultrasound is still used as a non-invasive method to enhance drug delivery, particularly for transdermal and localized drug delivery applications. 5. Liposome-based formulations. References 409, 411. Novel liposome-based formulations for prolonged delivery of proteins and vaccines were studied. Liposomes remain essential carriers for drug delivery, and research in this area continues to develop advanced liposomal formulations. 6. Biodegradable polymers. References 403, 434, 479, 498, 499. The references discuss various biodegradable polymers for drug delivery and tissue engineering applications. Biodegradable polymers are widely used today for controlled drug release and tissue regeneration. 7. Vaccine delivery. References 410, 461, 478. Some references focus on vaccine delivery and stabilization. Vaccine delivery systems continue to be a critical area of research, especially in the context of global health and emerging infectious diseases. 8. Electroporation and ultrasound for transdermal drug delivery. References 456, 457, 490. These references explore the combined use of electroporation and ultrasound for enhanced transdermal drug delivery. Such combination techniques are still being investigated for improving drug delivery across biological barriers. 9. Matrix metalloproteinases in wound healing. Reference 479. The study of matrix metalloproteinases in wound healing is relevant for the development of wound care and drug delivery systems aimed at promoting tissue repair and regeneration. 10. Polymeric controlled release vaccine delivery systems. Reference 484. Polymeric controlled release vaccine delivery systems were discussed. Today, there is ongoing research into improving vaccine delivery systems to enhance their effectiveness and stability. In summary, the references provided laid the groundwork for many areas of drug delivery and biomaterials research that are still highly relevant today. Advances in these fields have led to the development of innovative drug delivery systems and therapies for various medical applications. References 501 to 600. The references provided cover a wide range of topics related to drug delivery and tissue engineering. Here are some of the implications for drug delivery today based on these references. 1. Synthetic scaffolds in tissue engineering. Reference 501. The history of tissue engineering with synthetic scaffolds highlights the importance of designing biomaterials for tissue regeneration. Today, researchers continue to explore new synthetic materials and scaffolding techniques for drug delivery and tissue engineering applications. 2. Hepatocyte tissue engineering. Reference 502. Enhanced survival of transgenic hepatocytes is crucial for liver tissue engineering and drug metabolism studies. This research may impact the development of liver-specific drug delivery systems and by artificial organs. 3. Magnetically responsive liposomes. Reference 503. Magnetically responsive liposomes have the potential for oral drug delivery applications. This reference underscores the ongoing interest in innovative drug delivery methods that improve bioavailability and targeting. 4. Lyophilized compounds and pH memory. Reference 504. Understanding the pH memory of lyophilized compounds is valuable for designing sustained-release drug formulations. Today, researchers are developing advanced drug delivery systems that can modulate drug release based on physiological conditions. 5. Polyanhydride degradation. Reference 505. Studying the in vitro degradation characteristics of polyanhydride contributes to the development of biodegradable drug delivery platforms. These materials are relevant for controlled and sustained drug release. 6. Protein aggregation in polymers. Reference 506. Heterogeneity of serum albumin samples regarding solid-state aggregation has implications for sustained drug release from polymers. Researchers continue to investigate protein-polymer interactions for controlled drug delivery. 7. Polymer adsorption to microparticles. Reference 507. Understanding the adsorption of polymers to microparticles is essential for optimizing drug encapsulation and release characteristics. This knowledge informs the design of drug-loaded microcarriers. 8. Process simulation for protein production. Reference 508. Process simulation for recombinant protein production is relevant for biopharmaceuticals. Advances in this area help streamline the production of therapeutic proteins and peptids. 9. RGD peptid modified biomaterials. Reference 509. RGD peptid modified biomaterials offer interactive and resorbable properties. These materials are of interest for tissue engineering and drug delivery applications, especially for regenerative medicine. 10. Non-invasive drug release monitoring. Reference 510. Non-invasive monitoring of drug release and polymer erosion using techniques like EPR spectroscopy and NMR imaging is valuable for assessing the performance of drug delivery systems in vivo. 11. Zero-order drug release. Reference 511. Understanding zero-order release kinetics is essential for developing drug delivery devices that provide constant drug levels over time. This concept influences the design of long-acting drug delivery systems. 12. PEG-coated nanospheres. Reference 512. The development of PEG-coated nanospheres and their drug encapsulation properties has implications for nanoparticle-based drug delivery systems, which can enhance drug solubility and targeting. 13. Photo-cross-linked polymers. Reference 513. The synthesis and characterization of photo-cross-linked polymers are relevant for tissue engineering and controlled drug release. These materials provide platforms for local drug delivery. 14. Pulmonary drug delivery. Reference 514. Large porous aerosols for pulmonary drug delivery are an important area of research, impacting the treatment of respiratory diseases and systemic drug delivery through the lungs. 15. Controlled protein delivery. Reference 515. Controlled protein delivery from biodegradable microspheres is significant for applications in regenerative medicine and sustained drug release of therapeutic proteins. 16. Bioprosthetic heart valves. Reference 516. Novel approaches for constructing bioprosthetic heart valves have implications for cardiovascular drug delivery and tissue engineering of heart-related therapies. 17. Electrically conducting polymers. Reference 517. Stimulation of neurite outgrowth using electrically conducting polymers is relevant for neural drug delivery and tissue engineering of nerve-related disorders. 18. Film tension theory of phagocytosis. Reference 518. The film tension theory of phagocytosis provides insights into particle interactions with cells, which can influence the design of drug delivery carriers. 19. Surface reactive amino groups. Reference 519. Characterization of surface reactive amino groups on polymers is crucial for understanding biomaterial interactions, including drug delivery vehicle interactions with biological systems. 20. Immunogenicity of microspheres. Reference 520. Understanding the long-term immunogenicity of drug-loaded microspheres has implications for developing biodegradable drug delivery systems with minimal immune responses. These references collectively demonstrate the ongoing research efforts in drug delivery and tissue engineering, highlighting the importance of material science, controlled release strategies, and the development of innovative biomaterials for various therapeutic applications. Researchers continue to build upon this foundation to create more effective and targeted drug delivery systems. References 601 to 700. The references provided are from the late 1990s and early 2000s and cover a wide range of topics related to drug delivery and biomaterials. Here are some of the implications for drug delivery that can be drawn from these references. 1. Polymeric systems for controlled drug release. Reference 601. This paper likely discusses the development of polymers for controlled drug release. The implications include the potential for creating drug delivery systems that release medications at a controlled rate, improving patient compliance and reducing side effects. 2. Regional heparinization via simultaneous separation and reaction. Reference 602. This reference appears to relate to a novel method for regional heparinization. The implication here could be the development of improved techniques for anticoagulation during medical procedures or surgeries. 3. Cellular tissue engineering approaches. Reference 603. This reference suggests the exploration of cellular tissue engineering for medical applications. It implies the potential for creating functional tissues for transplantation or repair, including drug delivery within engineered tissues. 4. Rheological properties and microstructure of tissue-engineered cartilage. Reference 604. Understanding the rheological properties of tissue-engineered cartilage can aid in designing better drug delivery systems for joint-related conditions. 5. Transdermal photopolymerization of hydrogels. Reference 605. This reference likely discusses a method for creating hydrogel-based drug delivery systems through transdermal photopolymerization. The implication is the potential for improved transdermal drug delivery systems with precise control. 6. Transtissue photopolymerization of hydrogels. Reference 606. Similar to the previous reference, this one may discuss the photopolymerization of hydrogels for drug delivery. The implication is the development of new techniques for tissue-specific drug delivery. 7. Advances in skin electroporation. Reference 607. This reference implies advancements in skin electroporation, which can enhance transdermal drug delivery. The implications include improved drug delivery methods for various medical conditions. 8. Atomic force microscopy analysis of proteins. Reference 608. This reference suggests the use of atomic force microscopy for protein analysis. The implication is the potential for understanding and optimizing protein-based drug delivery systems. 9. Surface engineering and surface analysis of biodegradable polymers. Reference 610. This reference implies advancements in the surface engineering of biodegradable polymers. The implications include improved biodegradable drug delivery systems. 10. Tissue engineering of autologous aorta. Reference 611. This reference suggests the tissue engineering of autologous blood vessels. The implication is the potential for creating personalized drug delivery systems for cardiovascular diseases. 11. Poly, a eusic acid dimer anhydride. Reference 612. This reference likely discusses a specific polymer. The implication depends on its context but could relate to the development of new materials for drug delivery. 12. Protein stability in controlled release systems. Reference 613. Understanding protein stability in drug delivery systems is critical for biopharmaceuticals. The implication is the potential for more effective protein-based drug delivery. 13. Transdermal monitoring of glucose. Reference 614. Monitoring glucose levels through transdermal methods can have implications for diabetes management and continuous drug delivery. 14. Biodegradable photo-cross-linked poly-ester networks. Reference 615. This reference implies the development of biodegradable photo-cross-linked polymers for various applications, including drug delivery. 15. Tissue engineering. Reference 616. The reference to tissue engineering suggests the potential for creating tissues with integrated drug delivery capabilities for regenerative medicine. 16. Ultrasound-induced transdermal drug delivery. Reference 617. Ultrasound-assisted drug delivery methods can enhance drug penetration through the skin, leading to more efficient transdermal drug delivery. 17. Microchips as controlled release devices. Reference 623 Microchip technology can enable precise control over drug release, potentially leading to advanced implantable drug delivery systems. 18 Non-invasive drug delivery using low-frequency sonophoreses. References 631, 632, 633, 634 Low-frequency sonophoreses can be a non-invasive method for drug delivery. The implications include painless and efficient drug delivery techniques. 19 pH-responsive polymer microspheres. Reference 665 pH-responsive drug delivery systems can release drugs in response to changes in pH levels within the body, which can be useful for targeted drug delivery. 20 shape memory polymers. Reference 687 shape memory polymers can be designed for specific drug delivery applications, where the polymer changes shape in response to external stimuli, releasing drugs as needed. These references collectively highlight the significant advancements and potential applications of various drug delivery technologies and biomaterials during the late 1990s and early 2000s. These developments have likely had a lasting impact on the field of drug delivery, contributing to more efficient and patient-friendly drug delivery systems. References 701 to 800. The references provided cover a wide range of topics related to drug delivery and biomaterials. Here are some of the main implications and contributions of these references to drug delivery today. 1. Ultrasound-enhanced transdermal drug delivery. Reference 701. This reference explores the use of low-frequency ultrasound to enhance transdermal drug delivery. The study's findings have implications for improving the non-invasive delivery of drugs through the skin, which is a valuable approach for many therapeutic applications. 2. Skin permeability prediction. Reference 702. The research in this reference focuses on predicting skin permeabilities of various drugs, considering hydration effects. Such predictive models can aid in the development of transdermal drug delivery systems, helping to optimize drug formulations for skin penetration. 3. Neural stem cell seeding. Reference 703. This reference discusses seeding neural stem cells on polymer scaffolds. While not directly related to drug delivery, the use of scaffolds for cell-based therapies has implications for tissue engineering and regenerative medicine, including the potential for drug delivery in tissue-engineered constructs. 4. Reversible switching of surfaces. Reference 704. This reference explores reversible surface modifications, which can have applications in drug delivery, especially in the controlled release of drugs from surfaces or implants. The ability to switch surfaces on and off has implications for precise drug release control. 5. Microemulsion enhancers. Reference 705. The development of novel microemulsion enhancers for transdermal delivery of hydrophilic and hydrophobic drugs is significant. Such formulations can improve drug solubility and skin permeability, expanding the possibilities for transdermal drug delivery. 6. Low molecular weight heparins. Reference 706. Rational design of low molecular weight heparins with improved in vivo activity is relevant to anticoagulant therapy. These modified heparins may offer better pharmacokinetics and more convenient dosing regimens, potentially improving patient outcomes. 7. MEMS drug delivery devices. Reference 708. The biocompatibility and biofouling aspects of MEMS, microelectromechanical systems, drug delivery devices are crucial for their clinical viability. Ensuring that these devices are well tolerated in the body is essential for effective drug delivery. 8. Three-dimensional microfabrication. Reference 709. The ability to create three-dimensional microstructures, such as electrodeposited structures, has implications for various applications, including drug delivery devices. Precise microfabrication can enable the design of more efficient drug delivery systems. 9. 2-photon fluorescence microscopy. References 710 and 728. Visualization techniques like 2-photon fluorescence microscopy can be used to study drug diffusion pathways in tissues and across biological barriers, aiding in the design of targeted drug delivery systems. 10. Sustained drug release from microspheres. Reference 711. Decreasing the burst effect of drug release from PLGA microspheres is crucial for achieving sustained and controlled drug delivery. This reference explores strategies to improve the release profile of drugs from microspheres. 11. Gene delivery. References 715 and 716. The references discuss the synthesis and screening of degradable cationic polymers for gene delivery. Advances in gene delivery technologies are essential for various therapeutic applications, including gene therapy and personalized medicine. 12. Cardiac tissue engineering. References 717 and 751. Tissue engineering approaches for cardiac tissue and myocardial constructs have implications for drug testing and cardiac drug delivery. These models can be used to assess the effects of drugs on cardiac tissue in vitro. 13. Biodegradable polymer scaffolds. Reference 718. Understanding the degradation characteristics of biodegradable polymers, such as polyglycerol sabacate, is vital for the design of drug delivery systems based on these materials. It influences drug release kinetics and tissue integration. 14. Inhaled insulin. Reference 720. The development of inhaled insulin products has implications for improving patient compliance and convenience in diabetes management. This reference discusses recent advances in this field. 15. Nanocomposites for gene delivery. Reference 721. Polyhistidine PEG. DNA nanocomposites for gene delivery offer potential solutions for efficient gene therapy. These materials may enhance gene delivery while minimizing cytotoxicity. 16. Human embryonic stem cells on scaffolds. References 722 and 761. The use of scaffolds for human embryonic stem cell differentiation has broader implications for tissue engineering and regenerative medicine. It may eventually lead to the development of drug delivery systems incorporated into tissue-engineered constructs. 17. Microchip drug delivery. References 723 and 795. The development of microchip-based drug delivery devices allows for precise control over drug release kinetics. Such devices can be used for prolonged drug delivery in various therapeutic applications. 18. pH-triggered drug release. References 724 and 774. pH-triggered drug release systems are valuable for site-specific drug delivery. These references explore the design and performance of such systems, which can be applied to various drug delivery platforms. 19. Soft lithography and microfluidics. References 740 and 741. Soft lithography and microfluidics techniques enable precise patterning and manipulation of materials. These methods have applications in the fabrication of drug delivery devices and controlled release systems. These references collectively demonstrate the multidisciplinary nature of drug delivery research, spanning areas such as materials science, tissue engineering, microfabrication, and drug formulation. The findings and innovations from these studies continue to influence drug delivery strategies and technologies today. References 801 to 900. The provided list of references includes a wide range of research articles and reviews that span various aspects of drug delivery and related topics. While it's not feasible to provide a detailed analysis of each reference, I can offer some general implications and key takeaways for drug delivery research based on the information available in the references. 1. Diverse drug delivery mechanisms. Moss and Langer's work on drug delivery mechanisms, reference 801, highlights the importance of understanding the various methods and mechanisms involved in drug delivery. Today, researchers continue to explore and develop diverse drug delivery systems, including nanoparticles, microspheres, hydrogels, and more. 2. Biocompatibility and inflammatory responses. References 802, 803, and 804 emphasize the significance of studying the biocompatibility and potential inflammatory responses of drug delivery materials. Ensuring that drug delivery systems are well tolerated by the body remains a key concern in research today. 3. Targeted gene delivery. Reference 803 discusses the development of hierarchically self-assembling polymers for targeted gene delivery. Targeted delivery of therapeutic genes is an active area of research, aiming to improve the specificity and efficacy of gene therapies. 4. Nerve regeneration and tissue engineering. References 804, 818, and 826 delve into nerve guide materials, cardiac tissue engineering, and bone bioreactors, respectively. These studies reflect the growing interest in using drug delivery approaches to promote tissue regeneration and repair, which is particularly relevant in the fields of neurology, cardiology, and orthopedics. 5. Polymeric gene delivery. Reference 805 highlights structure/property studies of polymeric gene delivery. Developing safe and efficient gene delivery vectors remains a crucial focus in gene therapy research. 6. Smart materials. Reference 814 discusses smart materials with dynamically controlled surfaces. The development of responsive drug delivery systems that can release drugs in a controlled and triggered manner is an ongoing area of research. 7. Microfluidics and tissue engineering. References 848, 850, 851, and 883 emphasize the integration of microfluidics and tissue engineering. Microscale technologies and microfabrication techniques have become instrumental in creating complex tissue models and drug delivery systems. 8. Nanoparticles in drug delivery. References 869, 880, 883, and 897 highlight the use of nanoparticles in drug delivery. Nanotechnology has opened up new possibilities for targeted drug delivery and imaging, revolutionizing the field. 9. Aptamers for targeted drug delivery. References 869, 899, and 900 discuss the use of aptamers in targeted drug delivery. Aptamers, which are short DNA or RNA molecules, show promise in guiding drugs to specific targets in the body. 10. Biodegradable polymers. References 852, 868, and 874 emphasize the use of biodegradable polymers in tissue engineering and drug delivery. Biodegradable materials minimize long-term adverse effects and are crucial for controlled drug release. 11. Tissue-specific drug delivery. Reference 865 discusses the application of micro and nano-electromechanical devices for drug delivery. The ability to precisely target and release drugs in specific tissues or cells is a key focus. 12. Controlled release. References 867, 874, and 876 discuss the modulation of drug release within scaffolds and hydrogels. Achieving controlled and sustained drug release profiles remains a central goal. 13. Regulatory considerations. The references touch upon the complexity of regulatory approval for combination products, reference 854, and the need for understanding the safety and efficacy of drug delivery systems, reference 859. 14. Clinical translation. Many of these references provide insights into translational aspects, bridging the gap between fundamental research and clinical applications. In summary, drug delivery research today encompasses a wide range of strategies, including advanced materials, nanotechnology, smart delivery systems, and tissue engineering, all aimed at improving the precision, safety, and effectiveness of drug delivery for various medical applications. Researchers are also addressing issues related to biocompatibility, inflammatory responses, and regulatory considerations to advance the field further. References 901 to 1000. The provided list of references represents a wide range of research in the field of drug delivery and related topics, authored or co-authored by Robert Langer and his colleagues. While the references cover various aspects of drug delivery and biomaterials, here are some of the main implications for drug delivery research based on these references. 1. Nanoparticulate drug delivery. References such as 901, 904, and 911 highlight the use of nanoparticles for delivering drugs, genes, and therapeutic agents. This approach allows for targeted and controlled release, which can enhance drug efficacy while minimizing side effects. 2. Tissue engineering. References like 902, 915, and 964 demonstrate the importance of biomaterials and tissue engineering in drug delivery. Scaffold materials can be designed to support tissue regeneration and controlled drug release, which is crucial for regenerative medicine. 3. Neuroprotection. Reference 903 discusses neuroprotection using drug-loaded microspheres. This has implications for the treatment of neurodegenerative diseases and conditions. 4. Gene silencing therapies. Reference 906 mentions gene silencing therapies. This field has the potential to revolutionize the treatment of genetic disorders and other diseases at the genetic level. 5. Cancer therapy. References 905, 912, and 941 emphasize the use of targeted nanoparticles for cancer therapy. This approach can improve the specificity of cancer treatments while reducing damage to healthy tissues. 6. Microfabrication and microfluidics. References 907, 908, and 950 discuss microfabrication techniques and microfluidics, which enable precise control over drug delivery systems and allow for high-throughput screening of drug formulations. 7. Stem cell research. References 909 and 915 showcase the use of hydrogels and stem cells for tissue regeneration. This research has implications for drug delivery within regenerative medicine contexts. 8. Peritoneal adhesion prevention. Reference 910 addresses the prevention of peritoneal adhesions using a gel-based approach, which could improve surgical outcomes and reduce complications. 9. Transdermal drug delivery. References 965 and 966 discuss transdermal drug delivery, which has the potential to provide a painless and convenient route for drug administration. 10. Nanotechnology. References 954, 955, and 954 emphasize the role of nanotechnology in drug delivery. This includes the development of nanoparticles, nanoscale materials, and nanofabrication techniques to improve drug delivery systems. 11. Safety and quality control. References 974, 978, and 980 touch on safety and quality control in drug delivery. Ensuring the safety and purity of drug delivery systems is critical for clinical applications. 12 Clinical implications and adverse events. References 958, 961, 974, 975, 976 discuss real-world clinical implications, including adverse events associated with contaminated heparin. Such incidents highlight the importance of stringent quality control in drug manufacturing. These references collectively underscore the interdisciplinary nature of drug delivery research, where materials science, nanotechnology, biology, and clinical medicine intersect to develop innovative approaches for delivering drugs and therapies. The implications range from more effective cancer treatments to regenerative medicine breakthroughs and improved patient safety. References 1001 to 1100. The references provided cover a wide range of topics in drug delivery research, including nanoparticles, tissue engineering, gene delivery, and more. Here are some of the main implications for drug delivery research today based on these references. 1. Nanoparticle-based drug delivery. Several references discuss the development of lipid polymer hybrid nanoparticles. For example, reference 1001. These nanoparticles provide a robust platform for drug delivery. The implication here is that such nanoparticles can be tailored for specific drugs and applications, offering precise and controlled delivery of therapeutic agents, potentially reducing side effects and improving treatment outcomes. 2. RNA therapeutics. References like 1002 discuss therapeutic RNA interference, RNA, targeting specific genes. This approach has the potential to revolutionize the treatment of various diseases, including cardiovascular conditions and cancer. The development of safe and effective delivery systems for RNA molecules is a significant focus of current research. 3. Tissue engineering. References such as 1003 and 1008 highlight the use of innovative materials and scaffold designs for tissue engineering. The implication is that these technologies could lead to advancements in regenerative medicine, enabling the repair or replacement of damaged tissues and organs. 4. Protein adhesion. Reference 1004 discusses the study of protein adhesion and adsorption to microarrayed polymers. This research can have applications in drug delivery by improving the surface properties of drug carriers, allowing for better interactions with biological systems. 5. Small interfering RNA, siRNA, delivery. References 1005 and 1025 explore the delivery of siRNA to inhibit specific cellular processes. The development of safe and efficient siRNA delivery systems holds promise for treating a wide range of diseases by targeting specific genes or pathways. 6. Pharmacodynamics assessment. Reference 1006 addresses the assessment of siRNA pharmacodynamics in a living organism. This is crucial for understanding how drug candidates behave in vivo and optimizing their delivery for therapeutic effect. 7. Cancer therapy. Reference 1007 discusses nanoparticle delivery of suicide DNA for ovarian cancer therapy. Cancer treatment remains a significant focus of drug delivery research, aiming to improve the specificity and efficacy of therapies while minimizing side effects. 8. Polymeric materials for gene delivery. Reference 1009 discusses the synthesis of polyamino esters for gene delivery. Innovations in polymeric materials and gene delivery vectors are essential for improving gene therapy approaches. 9. Nanotechnology impact. References 1010 and 1064 discuss the impact of nanotechnology on drug delivery. Nanotechnology plays a vital role in the design of drug delivery systems, enabling precise control over drug release and targeting. 10. Barrier knockdown. reference 1011 discusses advances in siRNA delivery and knocking down barriers. Overcoming biological barriers is a fundamental challenge in drug delivery, and innovative strategies are being developed to enhance drug penetration. 11 microfluidic devices, reference 1012 describes the use of microfluidic devices for controlled drug delivery. Microfluidics offers precise control over particle formation and drug release, allowing for tailored drug delivery systems. 12 stem cell-based therapies, references like 1038 and 1048 highlight the importance of stem cell-based therapies and their delivery. Stem cell research combined with advanced delivery techniques holds promise for regenerative medicine and personalized medicine approaches. 13 biomaterials and hydrogels, references 1041 and 1046 discuss the use of biomaterials, hydrogels, and 3D scaffolds. These materials play a critical role in tissue engineering, providing a suitable environment for cells and drugs. 14 imaging and monitoring, references like 1039 and 1061 discuss the use of imaging techniques for monitoring drug delivery and cellular responses. Real-time monitoring and feedback systems are essential for personalized medicine approaches. 15 nanoparticle functionalization, reference 1021 discusses the formulation and preparation of functionalized nanoparticles for targeted drug delivery. Functionalization strategies enable the design of nanoparticles that can specifically target disease sites. Overall, these references collectively highlight the ongoing efforts to develop innovative drug delivery strategies, materials, and technologies that can lead to more effective and targeted treatments for a wide range of diseases. The field of drug delivery continues to evolve, offering exciting possibilities for improving patient care and outcomes. References 1101 to 1200. The provided list of references spans a wide range of topics related to drug delivery, biomaterials, and nanotechnology. Here are some of the main implications for drug delivery today that can be drawn from these references. 1. Biodegradable elastomers. Reference 1101. The use of biodegradable elastomers like polyglycerol sabacate can offer controlled drug release over time. This approach can be beneficial for sustained drug delivery, reducing the need for frequent administrations. 2. Core shell nanoparticles. Reference 1102. High throughput synthesis of core shell nanoparticles allows for the development of diverse nanocarriers for drug delivery. This approach enables the encapsulation of various therapeutic agents, improving drug stability and delivery efficiency. 3. Microfluidics for nanoparticle synthesis. Reference 1103. Microfluidic systems can provide precise control over nanoparticle size and composition, enhancing drug delivery system customization. This technology can be employed to create nanoparticles with tunable properties. 4. Nanotechnology in uro-oncology. Reference 1104. Nanotechnology plays a role in the field of interventional techniques, which can be applied to targeted drug delivery for urological cancers. This highlights the potential for personalized therapies in oncology. 5. Stem cell-based microvascular grafts. Reference 1105. Human embryonic stem cell-based microvascular grafts hold promise for cardiac tissue preservation and regeneration after myocardial infarction. This research suggests the potential for using stem cells in drug delivery and tissue engineering. 6. Fouling-resistant surfaces. Reference 1106. High throughput methods for discovering fouling-resistant surfaces can be beneficial for developing implantable drug delivery devices. Reduced fouling can lead to longer device lifetimes and improved drug release kinetics. 7. Real-time detection of reactive oxygen species. Reference 1107. Real-time detection of biomaterial-induced reactive oxygen species can inform the design of drug delivery systems, ensuring they do not trigger harmful immune responses. 8. Ultrasound-mediated transdermal drug delivery. References 1108 and 1112. Ultrasound-mediated drug delivery methods have the potential to enhance transdermal drug delivery. This technology can improve drug penetration through the skin, expanding options for non-invasive drug administration. 9. Enhanced transdermal delivery of nanoparticles. Reference 1109. Using ultrasound and enhancers like sodium lauryl sulfate can increase the transdermal delivery of nanoparticles. This could be significant for topical drug delivery applications. 10. Injectable thiolacrylate hydrogel. Reference 1110. Injectable hydrogels for sustained drug release, like thiolacrylate polyethylene glycol hydrogels, offer potential for long-acting drug formulations and localized drug delivery. 11. Engineering patent co-cultures. Reference 1111. Engineering systems for generating patent co-cultures can help control cell-cell interactions. This has applications in tissue engineering and drug testing, allowing the study of complex cell responses. 12. Responsive microgrooves. Reference 1115. Responsive microgrooves can be used to create harvestable tissue constructs. This approach holds potential for controlled drug delivery within tissue engineering scaffolds. 13. Intracranial microcapsule drug delivery. Reference 1116. Intracranial microcapsules can be used for drug delivery to treat conditions like gliosarcoma. These implantable devices offer targeted drug release in difficult-to-reach locations. 14. siRNA delivery and immune system. Reference 1117. Research on siRNA delivery and its interaction with the immune system is crucial for understanding the safety and efficacy of RNA-based therapies. 15. Sterilization of soft biomaterials. Reference 1118. Sterilization techniques for soft biomaterials are essential for maintaining the safety and integrity of implantable drug delivery devices. 16. Polymeric gene delivery. Reference 1135. Understanding the structure-activity relationships of polymeric gene delivery systems can lead to the development of safer and more efficient gene delivery vectors. These references collectively illustrate the interdisciplinary nature of drug delivery research, highlighting innovations in materials science, nanotechnology, microfluidics, and biotechnology. The implications for drug delivery today include improved control over drug release, enhanced targeting of specific tissues, and the development of personalized therapies. These advancements hold great potential for the future of healthcare and pharmaceuticals. References 1201 to 1300. The references provided cover a wide range of topics related to drug delivery. Here are some of the main implications for drug delivery based on the information in these references. 1. Biomimetic cell-penetrating peptids, CPPs. Reference 1201. Rational design of CPPs can enhance the delivery of drugs and therapeutic molecules across cell membranes. Improved drug delivery efficiency can lead to more effective treatments with lower doses and reduced side effects. 2. Microfluidic platforms for intracellular delivery. Reference 1202. Microfluidic devices offer a promising approach for intracellular drug delivery. Precise control over fluidic parameters can enable the efficient and targeted delivery of therapeutic agents to cells. 3 modular click-in emulsion nanogels, reference 1203. Nanogels can serve as versatile drug carriers with tunable properties. Bone-targeted nanogels hold promise for localized drug delivery to treat bone-related disorders. 4 benchtop fabrication of microfluidic systems, reference 1204. Improved fabrication techniques for microfluidic systems can enhance the development of advanced drug delivery platforms. Compatibility with various solvents is crucial for the versatility of microfluidic devices. 5 cell squeezing as a microfluidic intracellular delivery platform, reference 1205. Cell squeezing technology offers a robust method for intracellular drug delivery. This approach can be valuable for the delivery of various therapeutic agents into cells. 6 efficiency of siRNA delivery by lipid nanoparticles, reference 1206. Understanding the limitations of siRNA delivery by lipid nanoparticles is essential for optimizing RNA interference-based therapies. Addressing endocytic recycling challenges can lead to more efficient siRNA delivery. 7 analysis and prediction of defects in polymer microarrays, reference 1207. Quality control and optimization of drug delivery materials, such as polymer microarrays, are critical for ensuring consistent performance. Predictive models can help identify and address potential defects. 8 historical perspective on biomaterials and biotechnology, reference 1208. Biomaterials play a foundational role in drug delivery systems, tissue engineering, and regenerative medicine. Advances in biomaterials have significantly contributed to the field of drug delivery. 9 multiparametric evaluation of lipid nanoparticles for siRNA delivery, reference 1209. A comprehensive evaluation approach is essential for assessing the effectiveness of lipid nanoparticles in siRNA delivery. Multiparametric analyses can provide valuable insights into nanoparticle performance. 10 enhanced DNA delivery to the cytoplasm, reference 1210. Cross-linked linear polyethelenamine represents a promising approach for enhancing the delivery of DNA to the cytoplasm of target cells. These references collectively highlight the importance of developing innovative drug delivery strategies, optimizing carrier materials, and improving the efficiency and precision of drug delivery techniques. These advancements have the potential to revolutionize the field of medicine by enabling more effective and targeted therapies while minimizing side effects. References 1301 to 1400. The references provided include a wide range of research papers covering various aspects of drug delivery. Here are some of the main implications for drug delivery based on the information from these references. 1. Advancements in vaccine delivery, reference 1301 discusses the development of a mucosal vaccine against chlamydia trachomatis. This suggests that mucosal vaccine delivery strategies are being explored as a means to generate protective immune responses against infectious diseases. 2. Single injection vaccines. Reference 1302 talks about single injection vaccines, indicating efforts to simplify vaccination regimens. Such vaccines could enhance patient compliance and improve the efficiency of vaccination programs. 3. Nanoparticle hydrogels. Reference 1303 discusses the exploitation of electrostatic interactions in polymer nanoparticle hydrogels. This suggests the use of nanoparticle-based hydrogels for controlled drug delivery, potentially improving drug release kinetics and targeting. 4. Ultrasound-mediated drug delivery. Reference 1304 introduces ultrasound-mediated gastrointestinal drug delivery. This technique may offer a non-invasive and targeted approach for drug delivery, particularly for conditions like ulcerative colitis. 5. Gene silencing with dendrimers. Reference 1305 mentions dendrimeric siRNA for efficient gene silencing. This implies the development of advanced nanoparticle systems for delivering genetic therapies and gene-based drugs. 6. Cell-based drug delivery and therapy. Reference 1307 discusses non-genetic engineering of cells for drug delivery and cell-based therapy. This highlights the exploration of cell-based approaches for drug delivery and therapeutic applications. 7. Nanomedicines for endothelial disorders. Reference 1308 discusses nanomedicines for endothelial disorders, which may have implications for the treatment of cardiovascular diseases and related conditions. 8. Hydrogels with enhanced stability. Reference 1309 talks about covalent incorporation of trehalose within hydrogels for enhanced long-term stability and controlled release of biomacromolecules. This approach may improve the shelf life and efficacy of drug delivery systems. 9. Targeted nanoparticles for atherosclerosis. Reference 1310 introduces HDL-mimetic PLGA nanoparticles for targeting atherosclerotic plaque macrophages. This indicates efforts to develop targeted nanoparticle-based therapies for cardiovascular diseases. 10. Responsive hydrogels. Reference 1311 discusses injectable self-healing glucose-responsive hydrogels with pH-regulated mechanical properties. Such hydrogels have implications for diabetes management and responsive drug delivery. 11. Phototriggerable liposomes. Reference 1312 mentions repeatable and adjustable on-demand sciatic nerve block with phototriggerable liposomes. This highlights the use of light-responsive drug delivery systems for localized pain management. 12. Environmental applications. Reference 1313 discusses nanoparticles with photo-induced precipitation for the extraction of pollutants from water and soil. This research has implications for environmental remediation through drug delivery technologies. 13. Microfluidic antigen loading. Reference 1314 talks about microfluidic squeezing for intracellular antigen loading in polyclonal B cells as cellular vaccines. This suggests the development of novel methods for antigen loading in immune cell-based therapies. 14. Neutrophil responses. Reference 1315 explores neutrophil responses to sterile implant materials, which is important for understanding and mitigating the foreign body response to implanted drug delivery devices. 15. Gastrointestinal physiologic monitoring. Reference 1316 discusses physiologic status monitoring via the gastrointestinal tract, which has implications for real-time monitoring and drug delivery applications. 16. Biomaterials and molecular movement. Reference 1317 addresses the regulation of the movement of molecules by chemical materials, which is essential for designing effective drug delivery systems. These references collectively highlight the diverse and innovative approaches being explored in drug delivery, ranging from vaccines and nanoparticles to advanced hydrogels and cell-based therapies. These advancements have the potential to significantly impact the effectiveness and safety of drug delivery for various medical conditions. References 1401 to 1500. The references provided cover a wide range of topics in drug delivery and related areas of research. Here are some of the main implications and contributions from these references. 1. Reference 1401. This reference discusses the use of synthetic microparticles conjugated with VEF165 to improve the survival of endothelial progenitor cells. This approach could have implications for regenerative medicine and tissue engineering, as it addresses the challenge of enhancing cell survival and function in therapeutic applications. 2 Reference 1402. This study focuses on the role of Ly6C, low, monocytes in immunosuppression and resistance to anti-VEGF2 therapy in colorectal cancer. Understanding the mechanisms behind immunosuppression in cancer has implications for the development of more effective cancer treatments, including drug delivery strategies. 3 Reference 1403. The development of a humanized mouse model that recapitulates the fibrotic response to biomaterial implants is crucial for understanding and improving the biocompatibility of implanted medical devices and drug delivery systems. 4 Reference 1404. This reference discusses the applications of ethylene vinyl acetate copolymers, EVA, in drug delivery systems. EVA-based drug delivery systems have implications for controlled drug release and long-term therapeutic applications. 5 Reference 1406. Surface modification of membranes for retinal explants has potential implications for ocular drug delivery and treatment of retinal diseases. 6 Reference 1411. Circulating magnetic microbubbles for localized real-time control of drug delivery through ultrasonography-guided magnetic targeting and ultrasound offer a non-invasive approach to drug delivery, particularly in cancer therapy. 7 Reference 1412. The development of an oral once-weekly drug delivery system for HIV antiretroviral therapy is a significant advancement in improving patient adherence and the effectiveness of HIV treatment. 8 Reference 1415. Cellular scale probes for monitoring dopamine neurochemicals in a rodent model have potential applications in drug development and understanding neurochemical changes associated with various diseases. 9 Reference 1416. The review on smart biomaterials highlights the importance of developing advanced materials for drug delivery and tissue engineering, with implications for personalized medicine and regenerative therapies. 10 Reference 1425. The miniaturized neural system for chronic, local intracerebral drug delivery has implications for the treatment of neurological disorders and brain-related diseases. 11 Reference 1433. Advances in biomaterials for drug delivery are crucial for improving the effectiveness and safety of drug therapies, as well as for targeted drug delivery to specific tissues or cells. 12 Reference 1437. The development of bioresorbable composite polymeric implants with high mechanical strength has implications for the design of medical implants and drug delivery devices. 13 Reference 1446. Ingestible bacterial electronic systems for monitoring gastrointestinal health have potential applications in personalized medicine and the early detection of gastrointestinal diseases. 14 Reference 1451. Ingestible self-orienting systems for oral delivery of macromolecules offer a novel approach to drug delivery, with potential applications in oral immunotherapy and targeted delivery. 15 Reference 1453. Steerable microinvasive probes for localized drug delivery to deep tissue have implications for precision medicine and targeted therapies. 16 Reference 1458. The use of crystallized drug formulations to prevent implant fibrosis has significant implications for the development of long-lasting implantable drug delivery systems. 17 Reference 1459. The delivery of mRNA vaccines with heterocyclic lipids has implications for the development of next-generation vaccines and immunotherapies. 18 Reference 1467. a luminal unfolding microneedle injector for oral delivery of macromolecules opens new possibilities for oral drug administration and vaccination. 19 Reference 1468. A new approach for microfabrication of printed circuit boards with ultrafine traces could have implications for the development of miniaturized drug delivery devices and sensors. These references collectively highlight the diverse and evolving field of drug delivery, with potential applications ranging from cancer therapy and HIV treatment to neuroscience and gastrointestinal health monitoring. Advances in materials, technologies, and delivery strategies continue to shape the future of medicine and healthcare. The references provided cover a wide range of topics related to drug delivery and biomaterials. Here are some of the main implications and contributions of these references for drug delivery today. 1 Reference 1401, Ede et al. 2017. This study focuses on improving the survival of endothelial progenitor cells using synthetic microparticles conjugated with VEF165. The implication for drug delivery is the development of innovative approaches to enhance the effectiveness of cell-based therapies, which can have applications in regenerative medicine and tissue engineering. 2 Reference 1404, Schneider et al. 2017. This reference discusses the applications of ethylene vinyl acetate copolymers, EVA, in drug delivery systems. EVA is commonly used in the fabrication of controlled-release drug delivery devices. The implication here is the continued exploration and optimization of EVA-based drug delivery systems for various therapeutic agents. 3 Reference 1411, Chertok and Langer, 2018. This study introduces the concept of circulating magnetic microbubbles for localized real-time control of drug delivery using ultrasonography-guided magnetic targeting and ultrasound. The implication is the development of advanced, non-invasive drug delivery methods that can be precisely controlled, potentially improving the therapeutic outcomes of various treatments. 4 Reference 1412, Curtain et al. 2018. This reference presents the development of an oral once-weekly drug delivery system for HIV antiretroviral therapy. The implication is a significant advancement in patient compliance and convenience for long-term treatments, which can have broad applications beyond HIV therapy. 5 Reference 1422, Kang et al. 2018. This study discusses the design and synthesis of waterborne polyurethanes, which have implications for drug delivery materials. Waterborne polyurethanes can be used to create biocompatible drug delivery carriers and coatings for medical devices. 6 Reference 1433, Fenton et al. 2018. This review article discusses advances in biomaterials for drug delivery. The implication is that researchers are continually developing new biomaterials with improved properties for drug delivery applications, which can lead to more effective and targeted therapies. 7 Reference 1434, Tsung et al. 2018. This study presents the development of stabilized single-injection inactivated polio vaccine. The implication is the potential for improved vaccine delivery methods that can simplify vaccination programs and enhance global health efforts. 8 Reference 1440, Khan et al. 2019. This study discusses the delivery of endothelial siRNA in non-human primates using ionizable low molecular weight polymeric nanoparticles. The implication is the development of nanoparticle-based delivery systems for RNA therapeutics, which is a rapidly growing area of research. 9 Reference 1450, Verma et al. 2019. This reference presents an ingestible self-orienting system for oral delivery of macromolecules. The implication is the development of innovative drug delivery devices that can improve the oral delivery of biologics and other large molecules. 10 Reference 1466, Traverso et al., 2019. This study introduces an ingestible self-orienting system for oral delivery of macromolecules. The implication is the potential for transforming the way certain drugs are delivered, particularly for patients who require long-term treatments. These references collectively highlight the ongoing research and development efforts aimed at improving drug delivery methods, increasing patient compliance, and enhancing the therapeutic efficacy of various treatments. Advances in biomaterials, nanotechnology, and innovative drug delivery systems continue to shape the field of pharmaceutical sciences and medicine. References 1501 to 1561. The references provided cover a wide range of topics related to drug delivery and materials science. Based on these references, here are some of the main implications for drug delivery today. 1. Nanoparticle-based gene silencing. Reference 1502 discusses the use of nanoparticle-encapsulated cerners for gene silencing in the hematopoietic stem cell niche. This approach can have significant implications for the treatment of various diseases by targeting specific genes responsible for pathological conditions. 2. Brain interstitial fluid sampling. Reference 1503 presents a platform for microinvasive membrane-free biochemical sampling of brain interstitial fluid. This technology can provide valuable insights into brain health and disease and may facilitate the development of therapies for neurological disorders. 3. Drug delivery for traumatic brain injury. Reference 1508 discusses the delivery of siRNA in traumatic brain injury, aiming to overcome the blood-brain barrier, BBB, pathophysiology. This has implications for treating brain injuries and neurological diseases by delivering therapeutic agents directly to the brain. 4. Nanotechnology for infectious diseases. Reference 1509 highlights the role of nanotechnology in treating infectious diseases. Nanoparticles can be designed to deliver drugs or vaccines effectively, which is particularly relevant in the context of emerging infectious diseases like COVID-19. 5. Precision nanoparticles for drug discovery. Reference 1511 discusses the engineering of precision nanoparticles for drug discovery. This technology can accelerate drug development by delivering drug candidates to specific targets with high precision, reducing side effects and improving therapeutic outcomes. 6. Ingestible electronics for microstimulation. Reference 1506 introduces ingestible transiently anchoring electronics for microstimulation and conductive signaling. This technology has implications for personalized medicine and therapies involving precise electrical stimulation of specific areas of the gastrointestinal tract. 7. Exhaled aerosol analysis for COVID-19. Reference 1505 discusses the increase in exhaled aerosol in individuals with COVID-19. This research has implications for non-invasive diagnostics and monitoring of infectious diseases through breath analysis. 8. Nucleic acid delivery. References 1520 and 1540 focus on nucleic acid delivery for therapeutic applications. This includes mRNA delivery, which has gained prominence with the development of COVID-19 vaccines, and genome editing, opening new possibilities for precision medicine. 9. Mucus interaction in drug delivery. Reference 1537 explores mucus interaction to improve gastrointestinal retention and pharmacokinetics of orally administered nanodrug delivery systems. This is essential for enhancing the efficacy of oral drug delivery. 10 microneedle vaccine printing. Reference 1551 introduces a microneedle vaccine printer for decentralized manufacturing of thermostable mRNA vaccines. This technology can revolutionize vaccine distribution and accessibility. 11 immune stimulation with TLR agonists. Reference 1552 discusses the engineering of kinetics for toll-like receptor TLR, agonist release from prodrugs to enable tumor-focused immune stimulation. This has implications for cancer immunotherapy. 12 humanized mouse models. Reference 1553 describes the identification of a humanized mouse model for testing immune-mediated biomaterial foreign body responses. Such models are crucial for developing biocompatible materials for drug delivery and implants. These references collectively highlight the ongoing advancements in drug delivery technologies, including the development of novel nanoparticles, improved delivery methods for nucleic acids, and innovations in biomaterials for medical applications. These advancements have the potential to revolutionize the treatment of various diseases and improve patient outcomes. Book main ideas. These book references cover a wide range of topics in the field of controlled release, drug delivery, biodegradable polymers, tissue engineering, and more. Here are 20 main ideas from these references. 1. Controlled release systems. The books by Langer and Wise discuss various aspects of controlled release systems, emphasizing their medical applications and classification. 2. Biodegradable polymers. Chasen and Langer's book explores the use of biodegradable polymers for drug delivery, highlighting their significance in the field. 3. Angiogenesis. Steiner, Weiss, and Langer delve into angiogenesis, which is the formation of new blood vessels, and its importance in medical applications. 4. Biopolymers. Peppers and Langer's works in advances in polymer science focus on biopolymers and their relevance in various biomedical applications. 5. Liposomes in immunology. Cohen and Langer discuss the concepts and applications of liposomes in immunology, demonstrating their role in drug delivery and vaccine development. 6. Proteins and peptides delivery. Cleland and Langer's book explores formulations and delivery methods for proteins and peptides, critical in the development of biopharmaceuticals. 7. Tissue engineering. Lanza, Chick, and Langer's textbook of tissue engineering introduces the field of tissue engineering and its principles. 8. Synthetic biodegradable polymers. Attila, Mooney, Vacanti, and Langer discuss synthetic biodegradable polymer scaffolds, which are essential in tissue engineering and regenerative medicine. 9. Principles of tissue engineering. The multiple editions of the Principles of Tissue Engineering by Lanza, Langer, Vacanti, and Attila provide comprehensive insights into tissue engineering techniques and applications. 10. Polymer science in biology and medicine. Terrell and Langer's book on polymer science highlights the significance of polymers in biological and medical contexts. 11. Microfluidic technologies. D. Murthy, Kadam Hosseini, Langer, and Blander's book discusses microfluidic technologies and their role in advancing human health, including diagnostics and drug discovery. 12. Recent advances. Lanza, Langer, Vacanti, and Attila's 5th edition of Principles of Tissue Engineering likely covers the latest advancements and breakthroughs in the field up to 2020. These books collectively provide a comprehensive overview of key topics in the intersection of materials science, medicine, and biotechnology, emphasizing controlled drug release, tissue engineering, and the use of polymers and microfluidics in medical applications.
Purpose of these episodes not at all for advice or medical suggestions. These are aimed to provide support to peer pharmacists in training in educational and intellectually stimulating ways. Again, these are not at all for medical advice or medical suggestions. Please see your local board and state-certified health professional. The views of this podcast represent those of my guests and I.
Thanks for listening to the podcast series The New Student Pharmacist, where we discuss chemistry and pharmacy, as well as leaders in pharmacy careers, community, and chemistry and pharmacy research. We encourage you to support the work we are doing and follow us on Spotify and Apple Podcasts by subscribing for free. We are so glad that you were able to tune in today. Note, the views on the podcast represent those of my guest, and I take care and all the best.